Good morning, all, to the first Grand Rounds lecture of 2025. Our Grand Rounds lectures, for those who already know it, are sponsored by the Pathological Society and the Shanks Foundation. And we have these
We have to hear her.
usually within the middle of the month. And we have experts presenting their work. And the lectures are usually approximately 45 minutes. And then after that, we have the opportunity for Q&A. So it gives me great pleasure this morning to introduce Dr. Omer Bayraktar, who is based at the Wellcome Sanger Institute. He has a background history of doing fantastic work using single cell and spatial transcriptomics to characterize human brain cellular diversity in both health and disease. During his training as a scientist, he discovered neural stem cell patterning mechanisms, and this was published in Nature in 2013. And then he went on to describe the astrocyte layered diversity in the cerebral cortex, and this was published in Nature Neurology in 2020. His team is, as I mentioned, based at the Wellcome Sanger Institute, and he's developed cell to location computational tools to map the different cell types within the brain, and he'll be speaking about that today. So without further ado, Omer, I'll turn the podium over to you, and really looking forward to your talk this morning about cellular heterogeneity in glioblastoma. Thank you.
Thank you so much, Sarah. It's a huge pleasure and honor to be giving this lecture today. So really excited to connect with the society and tell you about our work in brain tumors. So as you heard from Sarah, my lab is really interested in understanding the fundamental pathobiology of human brain disorders using discovery-based single-cell and spatial omics tools. So I presume that most of you are familiar with these technologies. In this first slide, I just wanted to give you a bit of an overview of how my lab and others have been applying these to really decipher the pathology of the astonishingly diverse, neural cell types in the brain across different diseases. In lots of projects, sort of the first thing we do is to generate the list of parts. Can we actually identify all the human brain cell types in disease tissue samples as well as our controls? And for this, we use single cell omics-based technologies. The idea here is quite simple. We have a piece of tissue, we put it into a blender, we get a soup of single cells or nuclei. And, initially at the transcriptomic level, but now with the addition of chromatin accessibility and numerous other modalities, we're able to really comprehensively identify cell types and cell states, as well as disease-associated gene expression programs using these technologies. So that gives us a great sort of starting point to put together like the list of parts. But what my lab's really focused on over the past years is to put those parts together to spatially reconstruct a nervous system, so you can actually look into the pathologies with spatial resolution. You'll see throughout my talk that we do this using spatial transcriptomics, and a lot of the analysis that we do actually leverages the integrated power of spatial and single-cell transcriptomics using bespoke computational models. And this is really, we think, a fantastic starting point to sort of start digging into the pathological interactions across disease-associated cell types. There's a little diagram here, shows you, for example, how this approach can identify tumor microenvironment signaling into malignant cells and brain tumors that may be associated with phenotypic changes and other disease phenotypes in cancer cells. Today, I'd like to tell you about our largely unpublished work on understanding the fundamental biology of human glioblastoma. So hopefully you'll see this out in a few weeks, but it's a huge pleasure to sort of share the work we have done on glioblastoma with you over the past few years. So I think, glioblastoma, or GBM for short, needs very little introduction to this group. It's the most aggressive adult brain tumor. It's currently incurable, and it's a truly terrible disease. There are many challenges to treating GBM. And one sort of thing that I think everybody agrees on is that these tumors show an extensive amount of heterogeneity that has made it very difficult to pin down sort of the key pathological mechanisms in malignant cells. Uh, the sort of our inability to cure this tumor is really not for lack of trying. It's one of the best molecularly profiled solid tumors. And over now almost 15 years of research has shown that in these tumors, cancer cells exist across a diverse spectrum of transcriptomic cell states. So if you were to grab, for example, a tumor from a single patient and sort of look into the diversity of cancer cells with single cell transcriptomics, you can see that they exist across a transcriptomic spectrum that on one end resembles developing human brain cell types such as neural progenitors or oligodendrocyte progenitors, and on the other end they resemble cell types like cell types that are associated with mesenchymal-like properties that you normally don't expect to see in the brain. And sort of the prevailing hypothesis really in the field is that across different tumors, you have the combination of different genetic drivers as well as distinct cues from the tumor microenvironment that lead to different abundances of these individual cancer cell states. And those are then thought to give rise to these more sort of classically, historically defined tumor subtypes of GBM, such as the proneural neural subtypes being enriched for the neural progenitor-like states versus the mesenchymal-like subtypes enriched for for their respective state. And to make things even more complicated, these cell states are not static. So really beautiful animal modeling studies have demonstrated that cancer cells can actually zigzag and transition across these states. So for example, if you were to purify the neural progenitor-like states, cancer cells showing neural progenitor-like states from a tumor, xenografted into an animal model, it's been shown that those cells can actually give rise to all the other states. So at this point, you might really be wondering why I'm giving this talk, right? There's so much has been done on GBM. Again, it's one of the best molecularly profiled tumor. But the challenge really is making sense of all this data. While an incredible amount of work has been done, for example, on describing, the diversity of these cancer cell states, malignant cell states, we do not really understand these fundamental biology. So specifically, we do not really understand the biological significance of these cell states. Why should tumor cells mimic neural progenitors or, oligodendrocyte progenitors or, mesenchymal-like hypoxic cells? Second, how does this all come about? We have a pretty good idea of some of the key driver mutations behind these tumors, but we don't really understand how those mutations actually give rise to this phenotypic diversity of the cancer cell states. And finally, the plasticity of these cell states, the ability of the cancer cells to transition from one of these states to another is really fascinating. But how these states actually relate to one another and whether cancer cells sort of progress through tractable trajectories of these states that are conserved across every tumor is almost completely unknown. So a few years ago, interested in answering these questions, my lab with a really fantastic set of interdisciplinary collaborators, really spanning the full range from our clinical collaborators, the cell atlasing data generators, to computational data modeling folks. We started a new multimodal cell atlas of glioblastoma that we call GBM space. And this project was generally supported by the Wellcome LEAP organization. And the idea here was to really go blank slate. Can we actually sort of forget about all the different sort of malignant cell states, tumor subtypes that have been identified in GBM and try to sort of reconstruct the complexity of these tumors from scratch? And to do that, we really use the playbook that I showed you in the first slide. You can see here that we have applied, various cell atlasing technologies to sort of the most common form of these tumors, primary IDH wild-type tumors. This study was not the first application of any of these technologies, but we did sort of two things quite differently than other studies. So the first is we know that these tumors can have really extensive intratumor heterogeneity, right? They can unfortunately grow to nearly take up, like a half of a cortical lobe, um, uh, across different tumor sites within one patient. You can find really diverse biology of malignant and tumor microenvironment states. So to be able to really capture this, every single tumor that we profiled in this study was investigated using multi-site biopsies, including one last tumor that was profiled almost in its entirety across 15 different sites in a grid-like fashion. Second, as I told you at the beginning, we're quite interested in integrating all of these different single cell and spatial transcriptomic modalities. But we knew that these tumors had extensive intra and inter heterogeneity, right? We knew that certainly different tumors can have different genetic drivers that can lead to very different phenotypic states of cancer cells. So we did not want to sort of approach this like a normal cell atlasing project where you generate single cell data from one set of samples and spatial omics data from another set of samples. Instead, to account for the heterogeneity of these tumors, we ended up generating all these different modalities from serial tissue sections of every single piece of tumor, approaching almost 60 tumor blocks in the total project. And as you can see here, this was done quite deeply, capturing lots of cells, lots of tissue sections, really one of the largest, I think, cell atlasing projects we have done in Sanger to capture the biology of malignant cells. Now, in the lab, we are interested really in analyzing this data to look at multiple sort of aspects of GBM tumor biology. So that goes from understanding how these tumors are organized in space, and how this is linked to the cellular trajectories of the cancer cells. These will be sort of the 2 points that I'll focus today. But beyond that, we're also quite keen on studying the clonal organization of the states, the signaling from the tumor microenvironment, and also the transcriptional circuits driving these malignant cells. So in the interest of time, I'll have to focus on the first two, but if you're interested in the rest, please reach out. Now, in terms of sort of sharing our Atlas findings with you, we'll start with the single nucleus data. So I said at the beginning, we can use the single nucleus or single cell transcriptomics to identify in an unbiased way all the different cell types and cell states in our data set. And by digging into the transcriptomic profiles of over 1 million nuclei that we collected across 12 tumors, we were able to really finely annotate the different flavors of malignant as well as tumor microenvironment cell states. We're very interested in the tumor microenvironment. Again, in the interest of time, I'll have to ignore him in the rest of the talk. But you can see here that our atlas has really comprehensively captured all different types of major TME cells. We got lots of myeloid cells. They're, in fact, the most abundant cell in the GBM microenvironment. But also, we have lots of different kinds of neurons, oligodendrocytes, astrocytes, vasculature, and lymphoid cells. But our focus initially was really dig into the biology of the malignant cells. And by using a multi-pronged approach, we were able to recategorize the malignant cell states into 4 major groupings. So the first 2 of these groupings were quite similar to the cellular states people had described before. These were both developmental progenitor-like cell states based on the molecular programs as well as the pathways that were enriched in these malignant cells. The first ones clearly looked like oligodendrocyte progenitors or OPCs. So again, this is a neural progenitor that you can find both in the developing as well as in the adult brain. Here, obviously, these cancer cells are not proper OPCs. They are not, differentiating into oligodendrocytes, but, they are sharing lots and lots of molecular similarities with those cells. The second set of developmental like cancer cell states we identified here, ironically, even though this is a glial tumor, look like neuronal progenitors, right? So if I were to sort of go into detail, they look like almost like a neuronally committed radial glial progenitor cell. And you could see here, both at the sort of gene expression level, but also at the pathway level, clear enrichment of neuronal differentiation and synaptic pathways. The third major group that we identified, and the way we sort of described this group deviated from some of the sort of common terminology of these tumors. In this third group, we could see that malignant cells seem to exist across a spectrum that on one hand resembled astroglial cells of the brain. So, they kind of look like developmental homeostatic astrocyte-like populations. In the middle, they look like glial cells that were showing, enrichment of an injury response and wound healing program, which we and others describe as gliosis, a term that I'm sure a lot of neuropathologists are very, very familiar with. And on the sort of the opposite end of the spectrum, we could identify cancer cells that were showing clear, enrichment of hypoxia. So these are the cell states that have been described by others as astrocyte and mesenchymal-like. And throughout the presentation, I'll make it I'll go into greater detail to show you why we chose this terminology of gliosis and hypoxia. And last but not least, we could also identify cancer cells that were clearly proliferating based on their gene expression signature. Now, before I sort of tell you about how these different cancer cell states are organized in the tissue and how they relate to one another, I want to actually tell you a little bit about their distribution across the different tumors. So what you're looking at here is the sort of the abundances of all these different, both fine-grained as well as coarse-grained phenotypic classes of malignant cell states that we identified in our atlas. And you can clearly see here that some of these tumors are biased to have more of the gliosis hypoxia or developmental-like progenitors. But what we were really surprised to see is that even in those tumors, you were able to identify a little bit of the other cell types, demonstrating that these individual GBM tumors really span both developmental-like as well as gliosis and hypoxia cancer cell states, right? And that's going to be quite important to piece like the relation of these cell states to one another. The second thing is, I think I spent quite a bit of time describing to you why we did the multi-region biopsies. So we were quite interested to see if that would reveal indeed regional variation of these cell states. And when we looked into quantifying the most abundant cancer cell states per tumor site, we indeed saw really high levels of regional heterogeneity in these tumors. On average, there will be a single site in these tumors that would show a very, very different, phenotypic, enrichment of cancer cell states. And we were able to identify tumors that really showed almost like a completely distinct malignant cell state across each, tumor site, as, indicated with the little box here. So I think that really, suggests to us that the single-site tumor biopsies for GBM would be quite confounded to truly, stratify the phenotypes of the cancer cells as well as the subtypes of these tumors. And that's also going to be sort of quite important later in the talk in piecing this puzzle together. Now, the distribution of these cell states already tells us, sort of gives us a hint of the organization of the cancer cells in GBM. However, we were quite interested in understanding whether these distinct cancer cell states showed also distinct spatial organization in these tumors. And to be able to do that, we use the spatial transcriptomic technologies. I'm sure the society and all of you are now quite familiar with the diverse forms of spatial transcriptomic technologies. In this project, the one that we made heavy use of is described as spatially resolved sequencing. And while that sounds like a mouthful, the idea here is really to sort of read the full transcriptomes of tissues, whole transcriptomes with in situ spatial resolution using positional RNA capturing. So there are lots of different kinds of these technologies now, but they all share more or less the same approach of using a fancy microarray that has a bunch of positionally barcoded RNA capture oligonucleotides. You lay a thin piece of tissue on these arrays. These days, both FFPE as well as fresh frozen tissue samples are compatible with these. And really, this is just your ordinary type of tissue section that you would process for histology. Then you permeabilize the tissue samples, so all the transcripts snake out. They are positionally captured onto these arrays and then identified using next generation sequencing. So at the end of the day, the type of data we get from these assays really, gives you the sort of spatially barcoded positions of whole transcriptomes in tissue samples. These technologies have been around for a little over a decade now, but what's really kind of revolutionized their use in the community was the upgrades to their resolution, their spatial resolution over the past few years. And you can see here that some of the most sort of commonly used technologies here, such as the Visium spatial transcriptomics method from 10x Genomics or the slide sequencing method, offer much higher resolution than the previous versions of this tech. The commonly available methods really are the ones on the right here. You can see methods like StereoSeq allow you to go even deeper, really give you subcellular resolution. But if you wanted to apply these technologies in your lab, the type of stuff that you have access to easily and can run at scale would be the Visium technology that's shown here. As people who are very familiar with tissue biology, you might have noticed a problem here. Both the Visium and the slide sequencing method, the size of these, microarray spots or capture beats that positionally capture transcripts do not provide you true single-cell resolution. Visium with its 50 micron resolution, for example, would capture about 5 to 10 cells per location in most of mammalian tissues. So really, to get cell type specific information out of these technologies, you need computational approaches. So a few years ago, to be able to deal with this issue in collaboration with Oleg Stegel from the German Cancer Research Center and EMBL, we developed an algorithm that we call self-allocation. So this was the work of a fantastic graduate student, Vitaly Kleshchevnikov. And our idea here was to basically deconvolve and decompose non-single cell resolution spatial transcriptomic data into its constitutive cell types by using integration with single cell RNA-seq. I'm happy to go into the details of the model. We use a probabilistic framework to do this, but in brief, the idea here is using the single-cell RNA-seq data set as a cell type dictionary, where we can identify the average gene expression profiles of diverse cell states that is annotated in our reference. Then when we go into the spatial transcriptomic data sets with self-allocation, such as Visium, we are able to look into every single sort of spot or data point and computationally deconvolve the abundances of every single cell type annotated in the single cell reference. And this is what really sort of allows us in one shot to predict the spatial positions of diverse cell types and cell states identified in single-cell RNA-seq across the entire tissue samples. And it really sort of provides a starting point for higher level analysis of identifying tissue niches as well as cellular interactions between spatially co-localized cell types. Now, we're quite keen to apply self-allocation to GBM, and without further ado, I'll share the findings, but I'll start by saying that, in this project, again, we were quite sort of keen to preserve the heterogeneity, to account for the heterogeneity of these tumors. So when we did the integration of the single nuclei and the Visium data sets of our GBM tumors, we are actually able to integrate like and like. We took the single nuclei data set of each tumor and mapped it to its dedicated Visium data set using self-allocation. And what that showed was really a fascinating degree of spatial organization of these different cell states in these tumors. So what you're looking at here on the right shows you one of these sample Visium images. In the background, you can see the H&E of this tissue section, which is about 6.5 millimeters square in size. So it's the H&E gives you that sort of like anatomical, or organ, gives you an idea, gives you a handle on the anatomy of the tissue. And we'll talk about that a bit more in a few slides. And the sort of the little circles, the spots in the front are those 55 micron tissue locations, Visium spots, that are profiled in terms of their transcriptomes. And you can see here the different colors are the cell location predictions on the abundances of different cancer cell states. And you can see here is that even in this tiny, relatively small tissue section, 6 millimeter square tissue area, you can see this really striking segregation, spatial segregation of developmental-like and gliosis hypoxia cell states. Now, we could use this as a starting point to really quantitatively describe the relationships of these cell states to one another. But before that, I want to share with you, I think, a really peculiar observation, which further, I think, demonstrates how these spatial technologies could be useful. So we could, with the Visium cell location mapping, we could repeat the same sort of like cell abundance distribution quantification that we did with single nuclei. And if you were to sort of compare the abundances of different malignant cells identified by single nuclei sequencing versus the spatial mapping side by side per tumor here, you can see that the spatial mapping profiles suggest that the different tumors are perhaps a lot more similar to one another. With spatial transcriptomics, we were able to find sort of a lot more of the gliosis hypoxia cell states, which we think may be sort of biased against by the tissue dissociation required by single nuclei sequencing, given that these cells are really enriched in the hypoxic necrotic compartments of these tumors, and therefore are vulnerable to getting lost during the dissociation. So that, we think, is a really peculiar observation that suggests that these tumors might be a whole lot more similar to one another than we think. Okay. So we love looking at the pretty pictures of spatial transcriptomics, but we were quite keen to get a handle on quantifying the relationships of these different malignant cell states to one another in these tumors. So to do that, we were able to, at a tumor and patient level, we were able to first identify, the tissue niches underlying these tumors. This is a term that you hear a lot in tissue biology, and it's a term you hear a lot in spatial transcriptomics. I think different people load lots of different meanings into it, but the way we kind of interpret this simply is the groups of cells, which would be both malignant as well as tumor microenvironment cells, that are spatially co-localized in tumors in a recurrent manner, right? So it's a very, very simple quantitative definition. So we're able to do this at the level of each tumor, and then we're able to sort of cluster these tissue niches based on their shared cell state composition to see if different tumors share kind of the same architecture of the tissue niches. And what this showed really strikingly, that a small number of these recurrent tissue niches underlie the spatial organization of GBM. You can see here on the heat map here, shows the cellular sort of distribution of these different niches. As you can see, about 12 of these niches are observed in multiple patients, and they are distinguished from one another based on the enrichment of different flavors of malignant and TME cell states. Now, before I go into what these niches are, I want to share with you, I think, one of the most important findings of this line of analysis, which is really looking at the distribution of these malignant versus tumor microenvironment enriched niches across the tumors. So, when we did this study at the beginning, we were thinking that given the extensive GBM subtypes, tumor subtypes identified in the field, we might actually identify tissue niches that are enriched in just specific tumors and not observed in the rest. But really contradicting that, we were able to identify these niches, especially the ones that are defined by distinct malignant cell states, in almost every single tumor that we looked at in this study. So that really suggests that different tumors share this fundamental tissue architecture. And again, that will be quite key in piecing together how these different cancer cell states relate to one another. Okay. So an advantage of spatial transcriptomics is that once we define these kinds of properties, such as tissue niches, we are able to actually go back into the tissue samples and see the anatomical organization of these environments. And that really enables us to interpret them better. And when we did this in our tumors, we identified really that these niches are organized in a really sort of specific and stereotyped anatomical manner that represents the spatial transitions of malignant cell states from one another. So a bunch of you can probably already see from this image that, within the tumors, you have different compartments of these niches that are distinct from one another. You can see here, for example, that the developmental-like cell states, neuronal-like in this tumor, are right up against the infiltrated cortex where the tumors are expanding to, whereas the gliosis hypoxia states are further situated deeper in the tumor in the necrotic core. And again, this is something, we could interpret in a very sort of, quantitative, high certainty type of manner. So what you're looking at here now is taking all the Visium data we have generated from these tumors, from these 12 tumors, almost 100 sections, and quantifying in this graph the spatial proximity of these different niches. And what this really showed to us really was that there's almost like a single anatomical axis of these malignant niches. On one hand, you had the developmental-like cancer cell states that were enriched in the leading edge compartment of the tumors near the infiltrated cortex. And then progressively, as you move deeper and deeper, different flavors of malignant cell states showing astrocyte gliosis hypoxia phenotypes would appear. Now, this led us to ask a very specific question, and we'll again get to that in a few slides. But before we kind of progress with that, we really wanted to understand whether what we are seeing here, the spatial transcriptomic niche organization, would correspond to the known anatomical features of these tumors. So to do that, we worked with a fantastic NIHR academic clinical neuropathologist at that time, Melanie Jensen. And Melanie, with guidance and validation from both Annalise and May and Briggs at Adam Brooks Hospital, was able to do a comprehensive anatomical annotation of these tumors. So what you're seeing here now is Melanie investigating the H&E images of the Visium sections that we analyzed with spatial transcriptomics. And based on, the sort of the anatomical census of these tumors, annotating their different compartments just based on the histological information. And you can see here again, these tumors have a number of very distinct anatomical features. For example, the sort of the bulk of the tumor is identified as is sort of categorized as a cellular tumor compartment. But then as you go deeper into, the hypoxic and the necrotic regions, you can identify distinct spatial compartments such as the perinecrotic zone, the main necrotic area. But then there are also other anatomical features such as microvascular proliferation. So what Melanie was able to do really is to extend this annotation to almost half of the tissue sections from the variety of the different tumors that we analyzed. And that really enabled us to then sort of do really a comparison of our tissue niches versus the anatomical domains. And what you can sort of see here is that indeed the spatial organization we see with spatial omics really matches and sort of further enriches the known anatomy of these tumors. You can see, for example, that the developmental-like cancer cell states are located throughout the cellular tumor compartment that borders the infiltrated cortex. But then you can see that gliosis hypoxia states are really layered here across the deeper perinecrotic zone, pseudopalisading cells and necrosis, and necrotic areas, really suggesting that that the observations we made with spatial transcriptomics are, consistent with the anatomical organization of these tumors. And finally, I want to convince you further that the spatial organization of the tumors that we identified here is not something that, is not an artifact of, this fancy spatially resolved sequencing analysis and single cell data integration. So with the advance of new spatial transcriptomic technologies, we were able to use orthogonal approaches to validate our findings. And the one data type, new data type that we are quite excited about are imaging-based spatial transcriptomic methods. We find that these are quite complementary to sequencing-based technologies because while they're only limited to, say, few hundreds of genes, they actually give you true single-cell resolution using cyclic microscopy approaches. And what you can see here is us applying now xenium spatial transcriptomic technology to, to match tissue sections of these GBM tumors. And what we did here was to curate first a panel of genes, 315 of them, that include markers of different malignant and tumor microenvironment states. Then we're really able to measure the expression of these genes at single-cell resolution, and then see if the distribution of the malignant cell states matched what we saw in Visium spatial transcriptomics. And, what we sort of saw here was really highly consistent with our cell type and niche mapping results in Visium data. You can see here across 3 tissue sections or tissue sites of a given tumor, we are able to identify the enrichment of developmental-like cell states near the infiltrated cortex marked by non-malignant neurons. But as you moved further away from the infiltrated, the leading edge of these tumors, you started seeing these layers of gliotic and hypoxic-like cell states. So putting all that together, we thought that the organization that we are seeing here, where these different flavors of cancer cell states are organized really in this spatial manner, could be reflecting their sort of stereotype transitions in the tissue. And specifically, the idea we had here was perhaps, the organization here is really a sign of developmental-like cancer cell states progressively becoming a gliosis hypoxia states throughout tumor expansion. So looking at that in detail, we first dug into the compartments of these tumors that are marked by, gliosis and hypoxia. So as I told you before, from the single nuclei data, we were able to identify sort of a putative molecular trajectory of these cell states. And when we dug deeper, we could really clearly see here these gradients of gene expression in these cancer cell states. So on one hand, we had, developmental-like versions of these cells that express well-known markers of homeostatic astrocytes, such as GFAP, ALDH1-IL1, or GLAST. These became then sort of started to downregulate those homeostatic markers in the single nuclei data and started upregulating, the markers of markers associated with gliosis and wound healing. So these include well-known integrins, JAK-STAT signaling, annexins, and then, ultimately we could see that these cells were dominated by a classical hypoxia signature expressing genes such as VEGF-A, LPDA, and BNP3L. We could also see in the Visium data that these cells actually sort of showed this spatial transition from the cellular tumor that's bordering infiltrating regions where we see the enrichment of developmental-like cell states. We saw progressive sort of enrichment of these gliosis and hypoxia-like cell states in the different necrotic zones of these tumors. This was something we saw really clearly at the level of the spatial organization. So now, instead of showing you the distribution of the different niches, if I were to just plot the spatial positions of these different astrocyte, homeostatic astrocytes versus gliosis and hypoxia-like cell states, which we also quantify here with a graph, you see that they really demonstrate this layered organization in the tissue. So putting all that together, our hypothesis was really that the heterogeneity of these different cancer cell states here and the striking spatial organization might reflect a stereotyped trajectory of cancer cells from developmental-like states into those marked by injury response and hypoxia during tumor expansion. And in the sort of last sort of 5 minutes of my talk, I want to show you how we could actually test this directly using our data set. Now, single nuclei data really, or transcriptomic data really lends itself to the analysis of cellular trajectories, right? There are lots of different computational models that allow you to predict sort of the fates of different cells in single cell data. However, we thought it was quite important in this case to consider the clonal heterogeneity of these tumors. So we knew from previous studies that GBMs would have multiple phylogenetic. The cancer cells in GBM would consist of multiple phylogenetically distinct subclonal lineages, and it was really important for us to be able to sort of look into the trajectories of cancer cells within each clonal lineage. So that would sort of keep us from keep our analysis from being confounded by the genetic differences that could be distinguishing these subclones. And to do this, we took a number of different approaches, but briefly, one approach that we used was to, apply copy number inference to our single nuclei RNA and chromatin accessibility sequencing data. We are able to sort of apply this approach to both of these modalities, then computationally match the clones across them to come up with sort of like multimodal validated clones, as you may say. And what this really demonstrated was that first, we can indeed identify multiple clones per tumor. So what you can see here, based on the copy number plots across these different modalities, that we are able to really first, distinguish the malignant cells from the diploid tumor microenvironment. So you can see the chromosome 7 in amplification and the loss of chromosome 10 in all of these tumors. And all of these cancer cells. But then we are also able to identify copy number events that distinguish individual subclones, such as the alteration of chromosome 17 that you see that clearly distinguishes one subclone from the others. Now, the first question we asked here was that if there's a trajectory indeed from developmental-like into gliosis states, can you indeed see these cell states within each subclone? And the answer to this, as you can see here, was yes. Each of these individual subclones contained both developmental as well as gliosis-like hypoxia states. So that's sort of quite consistent with the trajectory idea. We did quite a bit of work on this, utilizing both spatial transcriptomics as well as spatially resolved whole genome sequencing using laser capture microscopy of tissue microbiopsies. So I'll have to sort of skip that in the interest of time. Sort of these studies really, I think, led to interesting sort of findings with regards to the spatial organization of these clones that we found to be very heavily intermixed. But one thing that I would like to sort of bring to your attention here is that while in each of these sort of different subclonal lineages, we were able to identify both developmental and gliosis hypoxia states, we were also able to identify really interesting differences across these lineages. So in this one tumor, you can see that in clone number 0, you have a different sort of flavor of developmental-like cell states. That clone is almost defined by neuronal-like cell states, opposed to the OPC-like developmental states observed in the other ones. So we think this is then a really, really interesting paradigm to start thinking about how the intra-tumor, genetic heterogeneity of these tumors, such as this chromosome 17 alteration, might be leading to these phenotypic switches. So that will be for another day. I'll just sort of continue with our trajectory testing. So, cutting a long story short here, once we demonstrated the shared clonal origins of these, different malignant cell states, we were able to look at their cellular trajectories using transcriptome-wide unbiased approaches. To do this, we used a new sort of cellular trajectory model that we recently developed with my collaborator Ole Stegel that we call Cell to Fate. And what you're seeing here is the application of this model to 3 subclones of cancer cells from one of these GBM tumors. You can see here the different colors show you the distribution of these cancer cell states across different points in the inferred cellular trajectories. The x-axis is really a directional trajectory. So the beginning of it is where the cells start out as, the early trajectory, the middle is the middle, and the end, the right side is the end of the trajectory. You can see here is that despite the sort of differential enrichment of developmental-like cell states across these clones, they each share the same trajectory where developmental-like states progressively turn into gliosis and hypoxia programs. And excitingly, when we extended this analysis to all of our tumors, we saw that this was the dominant sort of pattern of malignant cell trajectories in GBM. And I think quite strikingly, we saw that the tumors that were enriched for different flavors of malignant cells, such as tumors that you describe as proneural, neural, and classical mesenchymal, that are enriched for all these different cell states listed here, all showed the same trajectory, suggesting that this process, the transition from developmental-like states into gliosis and hypoxia, is really a trajectory that all GBM tumors have in common. So to summarize my talk, hopefully what you can see here, by leveraging the power of combined single-cell and spatial transcriptomic atlasing, we were able to identify a stereotyped cancer cell trajectory underlying glioblastoma heterogeneity across different subtypes of GBM, across different sort of currently defined subtypes of these tumors. We were able to identify that cancer cells exist on this transcriptomic spectrum from developmental-like to injury response and hypoxia. We were then able to demonstrate that these cell states are arranged in a very particular spatial manner in these tumors. And this relates to their conserved cellular trajectories, where they progressively transition from developmental-like cell states into gliosis and hypoxia during tumor expansion. So we're quite excited in this because in our mind, this takes the extensive heterogeneity of GBM that's been described to date and provides a new cell and tissue framework that allows us to relate all these different cancer cell states to one another, as well as different tumor subtypes. And a result of that, we also think here that the current nomenclature of what these cell states are and what these subtypes are needs to be revisited. Again, I think I talked in detail about why we think, cell states that have been described as mesenchymal relate to, cancer cells showing gliosis and hypoxia programs. But beyond that, we also think that some of the different subtypes of these tumors that have been described to date can also be confounded by previous studies that only looked at single tumor biopsies and sort of misinterpreted those tumors to consist of just those subtypes. So we think, for example, if you were to sort of dig back into the previous studies and look into tumors that were identified as neural and proneural, we expect that those were actually annotated. Those were annotated subtypes of tumor samples that were exclusively generated from the cellular tumor area near the infiltrated brain, missing the gliosis and hypoxia compartments of these same tumors. So in my last slide, it's been a pleasure to tell you about our work today. hope what you can see here, using sort of the power, the combined power of multimodal cell atlasing, we are able to tackle the extensive heterogeneity of these glioblastoma tumors and describe how this fundamental cell and tissue state trajectory unifies them. And we think this is also a really sort of exciting starting point now to think about how these tumors could be diversified further, right? So to think about how different patients can have different type of tumors, how these cancer cell states can show plasticity to break these targets, break these trajectories, or to identify therapeutic targets that would have a high chance of success across each tumor. We think this shared cell and tissue state trajectory is a wonderful starting point. So with that, I'd like to acknowledge, I think, my fantastic team, over the past 3 years. This has been a major project in the lab with contributions with many others. The work you saw today, it was mainly done by Tanya, was led by Tanya, Fani, and Grant. But beyond that, I'd also like to acknowledge our wonderful team of interdisciplinary collaborators, really spanning, a really amazing clinical cancer biology, computational biology group that's really made this happen. And last but not least, the patients who donated the patients, who gave us their tissue samples, as well as the Wellcome Leap organization that gave us very generous funding for this. With that, thank you so much for your attention.
Thank you very much, Omer. Just fantastic, fantastic talk. Huge data there, and just really succinctly and concisely presented. It was brilliant. We've already got two in the Q&A, so I'll start with those, even though I have a couple of questions myself. So from John Tedros, amazing work, and I'm a long-time C12 fan. Where do you see these technologies making first inroads into the clinic? And what do we need to make spatial G&T part of the diagnostic pathway?
Hello, John. Thank you so much. So fascinating question. So I think the technology enthusiast, if I put my technologist hat on, right, I'm of course very enthusiastic about the application of these techniques to the clinic. But I think we have to kind of, we have to be quite sober about what could be translated and what's practical to kind of translate. At the moment, I see really spatial transcriptomics mainly as a research tool that teaches about the fundamental biology of these tumors and can be applied in more clinical trial type of research settings, right? I think this also sort of this maybe perhaps slightly pessimistic look is also informed by, the complications of doing these things regularly in the clinic, right? And that goes from cost to having these really complicated, multimodal assays performed, performed routinely. So I don't really see in the near term that these technologies would become a part of the sort of clinical pathology diagnostic toolbox, to be completely honest with you. But I think they actually provide an amazing research opportunity to really dissect the biology of human disease. And I think this is also an amazing research opportunity between people applying these tools and pathologists. And, if you could sort of see there that the neuropathologists we work with were really key collaborators that helped us interpret that data. Now, in the medium to long term, though, I am optimistic about what could happen here. And I think there are few really, really promising applications. And the kind of things, just to be brief, I think the one application that I'm quite excited about is the ability to link spatial transcriptomic data into modalities that are routinely collected and accessible in the clinic. And that would be specifically the histology images. So you saw that in the Visium, as you demonstrated in your wonderful HypoMap study that recently got published. with the Visium, you're able to capture really like a decent H&E of the tissue section, as well as the spatial transcriptomes. So I think kind of a long-term promise is to build computational models that will really leverage large, huge amounts of this data with new architectures to be able to just look into the H&Es and predict the, spatial molecular properties. And I think once we have these types of foundational models, which again have been used in digital pathology to a certain extent, but not at this really almost single cell spatial resolution in the tissues, I think that could be transformative for the clinic.
That sounds fantastic if we were able to do that, everything from the H&E.
Yeah.
A question for Mark Ahrens. Very impressive and interesting talk. Which are the TME cells that you think may be important in interacting with the developmental, like GBM cells, them to change their state towards the hypoxic and gliotic states? And
Thanks so much, Mark. Wonderful question. So the TME is very near and dear to our heart, and sorry that I couldn't, I couldn't go into an interested time. We have found really striking specialization of the GBM tumor microenvironment to this trajectory of malignant cells from developmental-like states into gliosis and hypoxia. And specifically, what we have found is that first in these tumors, we're not the first ones to describe it, but I think we're the first ones to put it into an exact context that fits malignant cells. There is amazing heterogeneity of myeloid cells. So we can find in our single cell data lots of different flavors of resident, pro-inflammatory, infiltrating anti-inflammatory, as well as more recently described populations of hypoxic angiogenic stress response associated myeloid cells. And what was really striking is that if you, in our cell to location, in our spatial transcriptomic mapping, you're able to identify that these cell states showed almost like a mirror, mapped in an almost mirroring fashion to malignant cells. And specifically what we found was that the developmental-like malignant cells were neighbored by the combination of resident-like pro-inflammatory as well as infiltrating anti-inflammatory myeloid cells. Right. Then the gliosis and the hypoxia-like cell states, which are spatially distinct, were spatially co-located with this newly described populations of hypoxic angiogenic stress response associated myeloid cells. So that's, we think, a quite significant pairing of malignant cells and myeloid cells, but we were actually able to go beyond that, and we were able to sort of leverage the potential of single cell data to predict cell-cell communication. And we actually found that these spatially distinct myeloid populations provide distinct signaling molecules that target their neighboring malignant cells. So we think these signals from these diverse myeloid populations play a huge role in the progression of cancer cells across this trajectory.
That's great. I'm going to squeeze in one of my questions, if I'm allowed.
Yeah, please.
So the brain samples that you've been analyzing, do you know if they're treatment naive, or have the patients been treated with either radiotherapy plus or minus chemotherapy? Do you know that information?
Yes. So basically, treatment naive. So so GBM, unfortunately, is more or less a surgical tumor. So what we wanted to start with was really the primary tumors that, that in our mind, really, that's what you need to sort of get rid of ultimately. And what really happened for our tumors and other IDH wild type tumors in the field is that patients would get diagnosed with a GBM, so they'll get the brain scans after they show up at the clinic with really bad headaches and so on, other kinds of like symptoms. And then in a month, they are in the theater, they are getting the tumor resected. So during that period, most patients will get a steroid treatment. So in that way, all of our tumors have been lightly treated with that. But the chemo and the radiotherapy, the temozolomide and the radiotherapy happen after the surgery. What's really interesting to us is that there's been a lot of studies now that do the recurrent versus primary GBM comparison, right? Because there's quite a bit of interest in understanding is there something that changes in the recurrent tumors that leads to worse outcomes. And a lot of the studies are now suggesting that there is not that much change in the malignant cell states, and perhaps it's the tumor microenvironment that changes more using cell atlasing. However, we can already tell from the data that others have generated that the recurrent tumors do not have the same spatial organization.
Mhm.
So to us, that's quite interesting. You know, those tumors are obviously treated quite heavily. It's possible that, right, the tissue architecture is blasted away by the chemo and the radiotherapy, but there's something interesting going on in the recurrent tumors where I think the cancer cell trajectories and their tissue architecture are uncoupled.
Right. That's great. Unfortunately, we're going to have to leave it there. It was, fantastic talk, Omer, and um, I just encourage everybody who hasn't had the chance to ask you questions that they reach out to you, and also to remind everybody that these talks are recorded and would be available on PathSoc's website. And then last but not least, just to remind everybody that we will have our next month's lecture, and we don't have we haven't been able to tie down a speaker as yet. However, if you could mark your diaries for next month, and just stay tuned to this great series of Ground Round lectures. Thank you very much, and have a nice day.
Thank you very much.
Bye.